This is Rajendra. Today we are going to discuss about photosynthesis part 3, that is light reaction as well as dark reactions. In the process of photosynthesis, there are two important phases, that is a light reaction as well as dark reaction. In the light reaction, water molecule, sunlight, chlorophyll are used for synthesis of chemical energy, that is ATP as well as NADPH2 along with oxygen. ATP and NADPH2 form of energy is used in the dark reaction for synthesis of starch. First we try to understand light reaction process. Generally, light reaction takes place in the chlorophyll, in the sense chloroplast. In the chloroplast, there is a grana and stromal regions are present, but the light reaction takes place in grana thylakoids. In the light reaction, water molecules split into electrons, protons and oxygen. The electrons have been moving through various electron acceptors for synthesis of ATP form of energy, NADH2. Formation of energies takes place by the help of protons which is released from water molecule. First we can try to understand the stages of light reaction. The first one is an cyclic electron transport as well as non-cyclic electron transport. What's the importance of these two phases in this process? Light energy is used, chlorophyll molecules are used, as well as then ATP form of energy is produced, as well as NADPH2 form of energy is produced. First we can try to understand cyclic electron transport. First we can try to understand cyclic electron transport. It is in this process, electrons have been moving through electron acceptors. At last, ATP form of energy is produced. Where the electrons are starting, the electrons are written to the same component, hence is called as a cyclic electron transport. In this process, the ultimate is an ATP formation of energy. The ATP formation of energy is used in the dark reaction, which is going to take place in the grana, in the stromal thylakoids. Now we can try to understand first that already you know that there is a photosystem 1. In the photosystem 1, there is a light harvesting center as well as reaction center is present. The chlorophyll molecule, that is a chlorophyll A, where does reactions takes place? Called as a reaction center. Around the chlorophyll A molecule, that like chlorophyll B as well as carotenoids, that chlorophyll B as well as carotenoids, which are using for harvesting of light, called as a light harvesting complex. Light harvesting complex as well as reaction center all together is called as a photosystem 1. In the photosystem 1, P700 nanometers wavelength light is captured by light harvesting center and handed over to the reaction center. That is nothing but chlorophyll A. Then whatever the electrons are present in the chlorophyll A is excited in the presence of P700 nanometers wavelength light. The P700 nanometers wavelength light is useful for absorption, for splitting, for excitement of electrons. Now electrons are excited and moves away the chlorophyll A molecule, and these electrons are absorbed by electron acceptor, that is a pyridoxine, primary electron acceptor. That electron acceptor is convey the electrons to the one more electron acceptor, that is a pyridoxine. Now electrons or acceptors are moving to the cytochrome B6, then moves to the cytochrome F, and they convey to the plastocyanine. Means electrons are not stable. Electrons are not sticking with electron acceptor, any electron acceptor. Electrons movement is started from reaction center, that is a chlorophyll A, to primary electron acceptor, and the electrons have been moving to the different electron acceptors. When the electrons are moving to the cytochrome B6 as well as cytochrome F, then protons can move from one side to another side. That is nothing but from stroma to the lumen region. Because of this photosystem, there is nothing but cyclic electron transport takes place. The thylakoid membrane, one side is the stroma is present, the inner side is the lumen is present. Stroma is present, the inner side is the lumen is present. When the electrons are moving from one to another direction, protons are trying to move from outside to inside, in the sense from stroma to lumen region. When the protons are moving, ATPase is activated. ADP plus inorganic phosphate together form as an ATP. In the sense, when the electrons have been moving one to another, Protons movement is possible from stroma to the lumen region. Hence, ATP formation is possible. In the sense, in the cyclic electron transport, the reaction center is chlorophyll A. The absorption of light is in P700. Electron movement is through the electron acceptor. The source of energy formation is possible only ATP. NADPH2 formation of energy is not possible in the cyclic electron transport. Later we can see the non-cyclic electron transport. Then second phase of the light reaction is non-cyclic electron transport. In the sense, electron transport is starting from one point, ending with one other point. Electron does not return, but in the cyclic process, electrons where they start, they will be returned to the same point. But in the non-cyclic electron transport is starts from the PS means photosystem 2 and ending with the photosystem 1. In this process, photosystem 1 is consist of chlorophyll A, where the reaction center is called, as well as light harvest center, which is the chlorophyll B, as well as carotenoids. All together is called as the light harvest center. Light harvest center used for absorption of light and hand over to the reaction center. Now the reaction center have the capacity for absorption of P680 nanometers wavelength light of red color. When the light is absorbed by the chlorophyll A, the electrons will be excited. Where the electrons are getting here. In this non-cyclic electron transport, source of the electrons are nothing but from water. Water molecules are splitting into protons as well as electrons as well as oxygen. From each water molecule, 2 protons, 2 electrons, half water oxygen is liberated. This oxygen is liberated in the atmosphere as a source of oxygen, but these protons and electrons are accumulated within the lumen part of chloroplast. In these electrons, whatever the water molecule splitted, from the splitted water molecule, electrons are getting outside as well as protons. In the sense, in the light harvesting, in the sense photosystem 2 have the capacity for splitting a water molecule. That P680 nanometers wavelength light, that is nothing but photosystem 2 act on water molecule for splitting of water. This water is get split as a protons, electrons and oxygen. Oxygen liberates into atmosphere, protons and electrons remains within the lumen part of chloroplast. Now look at here. These electrons which is coming from water is a source for electrons excitement and for the movement of electron transport through the electron acceptors. Does that mean these electrons are coming from water splitting? One concept. When the light is passing on here, that light is out on the water for splitting. Then electrons comes out. These electrons are absorbed by the chlorophyll A. Now electrons are get the excitement. These electrons are excited and captured by primary acceptor of electron, phosphyte. Now electrons are not stable. Electrons are not sticking with any electron acceptor. It is moved to the one more electron acceptor, that is a plastoquinone. And the plastoquinone to electron acceptors move to this. Electrons are moving to that one more electron acceptor, cytochrome B6, and to that cytochrome F. When the electrons are moving to plastocyanine, means when the electrons are moving in between cytochrome B6 and cytochrome F, that protons are possible to move from one side to another side. In the sense, from stroma to the lumen part. Example is the chloroplast structure. It is a membrane structure. Outside is the stroma is present. Inner one is the lumen present. When the electrons are moving to the cytochrome B6 and cytochrome F, protons are moving from stroma to the lumen side. When the protons are moving, means when the protons are moving from the stroma to the lumen side, there is a ADPase. ATPase is activated. As per the protons movement, ADP plus inorganic phosphate together is formed as ATP. In the sense, in the non-cyclic electron transport, from PS means photosystem 2 photosystem 1, in between photosystem 2 to photosystem 1, ATP formation is possible with the help of protons movement, with the help of ATPase enzyme. At the same time, now electrons are handed over the photosystem 1. In the photosystem 1, same reaction center is present, there is a chlorophyll A. Light harvest center is present, light harvesting complex is present, other than chlorophyll A molecules. Now here, P700 means 700 nanometers wavelength light is captured by reaction center. Now electrons are excited. From the way the electrons are getting from the photosystem 2, means itself photosystem 1 itself do not have the electron production. Whatever the electrons are given from the photosystem 2 by the splitting of water molecule, now the electrons are excited and it is accepted by ferredoxine system. In the sense, primary electron acceptor. Now electrons are moving to the two electron acceptors of PS1, that is the ferredoxine. When the ferredoxine have the electrons, there is the enzyme is present here, NADP reductase. When the NADP what is the difference between cyclic electron transport, non-cyclic electron transport. And here is, in the cyclic electron transport, only photosystem 1 is involved, but in the non-cyclic electron transport, photosystem 1 and photosystem 2 are involved. In the photosystem 1, there is a reaction center have the capacity for absorption of only P700 nanometers wavelength light. But in the photosystem, in this one non-cyclic, photosystem 2 is present, photosystem 1 is present. In the photosystem 2, reaction center chlorophyll A have the capacity for absorption of P680 nanometers. In the photosystem 1, P700 nanometers wavelength light. Means two reaction centers are participating, then two photosystems are participating, the two chlorophyll molecules are participating, two different wavelength light can be observed, capturing is possible. In this cyclic, second difference is utilization of only longer wavelength of light P700, that is only P700. But in this non-cyclic, using both longer P680 as well as shorter wavelength light of P700, 2 is possible here. Then here electron moves in closed circle. Here you see that electron's movement is starting from the reaction center and move through the across electron acceptor, and at last electrons are moving back to the same reaction center. But in this non-cyclic, electrons movement is starting. Electrons moves in the zigzag manner or Z scheme. Zigzag in the sense electron movement is starting from photosystem 2 and moves across this electron acceptors and hand over the photosystem 1. Again the moves to the across the electron acceptors. When the movement is starting from the photosystem 2 to the PS2 as well as electron acceptors. At last these electrons are handed over to the NADPH. In the sense NADP plus H plus in the sense protons as well as electrons together is formed as an NADPH2. In the sense electrons are at last used for the formation of NADPH2, another form of energy in photosynthesis process. This and photolysis of water does not take place here. In this process, splitting of water does not take place. The liberation of oxygen is not possible, protons liberation is not possible, electron liberation is not possible. In this process, electrons which is given from photosystem 2 is only going on here. But in this one, what is oxidized means what is oxidized and splitting into the protons, electrons and oxygen. Oxygen is liberated into atmosphere. Protons are using for high concentration, you move from high concentration to low concentration from formation of ATP. These electrons also moving through the electron acceptor. At last, electrons are handed over to this NADPH. Then here only ATP formation is possible. In this one, ATP formation is possible in between that cytochrome B6 and cytochrome F. But here is a ATP formation is possible as well as NADPH formation is possible. ATP formation is possible in between the cytochrome B and cytochrome F. NADPH2 formation is at end of this one electron acceptor of photosystem 1, that is a paradoxine. At last, NADP reductase enzyme is used for the formation of NADPH2. Now we can try to understand that chemiosmotic hypothesis. It is one of the important concepts. Based on this concept, we can try to understand that formation of ATP and formation of NADPH2. How does ATP formation takes place through ATPase enzyme, and how does NADPH2 formation takes place with the help of NADP reductase enzyme. In this chemiosmotic process takes place in the thylakoids, that is an granum thylakoids. It is a membrane. It is like a one membrane system. Like this, many membranes together is called as the stalk of the membranes. That's the thylakoids. In this membrane, outer surface, lower surface. Outer surface is called as a stroma. The inner area is called as a lumen. Means we are talking about stroma, membrane system, lumen portion. These are three areas we are talking here. This is called as a membrane. Among these three, what changes are going on here? What the process going on here? This theory can explain ATP formation process, NADPH formation process. This theory also explains the connection between the photosystem 1 as well as photosystem 2. In this one, now first you can look at the non-cyclic photophosphorylation. These are non-cyclic photophosphorylation. In this process, here there is a reaction center is present, chlorophyll A the chlorophyll A can capture, along with light harvesting complexes are present, they can capture P680 nanometers wavelength light, that is a red light captured by this pigment, and this light is this light is effect on the water molecule. At the time of photosynthesis process, plant need water molecule, chlorophyll as well as light, along with the light carbon dioxide. But in the light reaction, in the chemiosmotic process, light is utilized, water molecules are utilized, as well as chlorophyll molecules are utilized. Except carbon dioxide, remaining 3 factors are participating out of 4 factors. In this process, light is captured by PS2, photosystem 2. That light is effect on the water. Then water molecules are splitting into electrons, protons as well as oxygen. From each 2 water molecule, 4 protons as well as 4 electrons comes out, along with 1 molecule of oxygen. This oxygen is liberated into atmosphere, and these protons, electrons are present within the lumen because of its lumen portion. And these protons not accepted by any proton acceptor, means not at all present any proton acceptor. But electrons are received by electron acceptors, that is an ferredoxine, cytochrome F, cytochrome B, so on. But protons are not accepted by any proton acceptor. Proton acceptor is not present. Then from time to time, water molecules is splitting. The light is absorbed by this photosystem 2. Then it impact on the water molecule. Water molecules have been splitting. After some course of time, the protons concentration have been accumulated within that lumen area. But these electrons are passing through this electron acceptor. Then the protons concentration have been accumulated. The concentration of proton within the lumen area is increased. Then protons will try to move from high concentration to low concentration. Where is the low concentration? In the lumen area, protons concentration is more, but in the stroma area, protons concentration is less. Hence, the protons try to move from lumen to stroma region. Then this membrane does not accept the protons move in any place, every place. No, protons can be possible to move through only ATPase enzyme. On this thylakoid membrane, a special ATPase enzymes are present. Wherever the ATPase enzyme is present, the protons possible to move from high concentration to low concentration. This ATPase enzyme consists of 2 parts. One is a CF0, one is a CF1. Means coupling factor 0, coupling factor 1. CF0 is embedded in the thylakoid membrane, in the sense completely within the thylakoid membrane, but CF1 is out of this thylakoid membrane, that is close to stromal region. Means 2 portions are present. CF0, CF1. CF0 is called as a stalk, CF1 is called as a head. Head part is always towards stroma region, the stalk part is always towards, then on the surface of, in the middle of thylakoid membrane. Through this ATPase enzyme, protons are moving from high concentration to low concentration. When the protons are moving from high to low concentration, ADP plus inorganic phosphate together is react to form as ATP. In the sense, ATP formation takes place with the help of ATPase enzyme, especially based on concentration gradient of protons. These protons have been moving from lumen to out of this lumen, that is a stromal region. In the sense, one direction is one. One other side is electrons also produced by splitting of water. These electrons are accepted by primary electron acceptor. Then it is passed into that electron acceptors, plastoquinone as well as cytochrome B, cytochrome F. These electrons are moving to plastocydine. When the electrons are moving to the plastocydine, these electrons are handed over to the PS1, photosystem 1. As per the photosystem 1, electrons are moving to the electron acceptors, that is a ferredoxine. Now the one point is, when electrons are moving from PS to the PS1, we have discussed already ATP formation is possible. How does it possible? Now, when the electrons are moving from cytochrome B to cytochrome F, some amount of protons are moving from stroma to the lumen region. How? When the protons are moving from lumen to out of this lumen, stromal region, ATPase enzyme is activated, ATP formation is possible. When the protons are moving from inside to outside, the accumulation of protons out of this lumen, means within that stroma, takes place. These protons want to move from high concentration to low concentration. When the protons want to move from high concentration to low concentration, they also can choose ATPase enzyme. This ATPase enzyme is present, means there is an in between cytochrome B and cytochrome F. When the electrons are moving, protons are accepted to move from high concentration to low concentration. When the protons are moving, as a discussion here, ATP formation is possible in between cytochrome B as well as cytochrome F. In the sense, ATP formation is possible in between cytochrome B, cytochrome F, that is in between PS2 and PS1, as well as ATP formation is possible through the ATPase enzyme. When the protons are moving from high concentration to low concentration and high concentration to low concentration. Initially moving from the lumen to stroma, later stroma to lumen. So the concentration gradient helps for the formation of ATP. In this concept, then first concept is the formation of ATP as well as formation of NADPH. Two types of energy is formed. The two types of energy is going to use in the dark reaction. In this, some of these important points are here. The first one is movement of protons from high concentration to low concentration based on the concentration gradient. That is nothing but protons, which is produced by splitting a water molecule, moves from high concentration to low concentration. Then splitting of water takes place here. In the sense, water molecule splitting into that electrons, protons and oxygen. Without water molecule splitting, photosynthesis does not take place. Then photosynthesis takes place with the help of splitting water molecule. The splitting of water molecule is also called as photolysis. Then each water molecule gives rise to 2 protons, 2 electrons and half oxygen. The 2 water molecules are participating, 4 electrons, 4 protons as well as 1 oxygen molecule. It is in the presence of light as well as chlorophyll. Then third concept is ATPase is activated for the formation of ATP. In the sense, ATPase enzyme is present for the production of ATP. Then here ADP plus phosphate, inorganic phosphate, together is formed as ATP. ATP in the sense adenosine triphosphate. ADP in the sense adenosine diphosphate. Inorganic phosphate. Adenosine diphosphate is added with one more inorganic phosphate, together is formed as an adenosine triphosphate. Triphosphate. That's it. This formation is possible in the presence of ATPase enzyme. Then one more is an NADPH reductase enzyme is present in the Stroma region. It is help for the formation of NADPH2s. In the sense, NADP is an acceptor of proton along with the protons present here. Together is formed as NADPH plus H plus, and a final product is an NADPH2. This conversion is possible with the help of NADP reductase enzyme. This question was asked in the past NEET exam. Which enzyme is used for the formation of NADPH2 from NADP plus protons 2H? Then concept is the structure of ATPase. ATPase enzyme consists of 2 components. One is an CF0, which is embedded in the thylakoid membranes. The one more structure is an CF1, then which is out of this thylakoid membrane, that is a Stroma site, that is CF1. So coupling factor of 0, coupling factor of 1, that is the structure of ATPase. Coupling factor of 0 is also called as a start. Coupling factor of 1 is also called as head part. Along with that, this chemiosmosis process requires some conditions. Then basically 4 conditions are important. This question also asked in the past. The first condition is a membrane is important. Which membrane here? Thylakoid membrane is important for chemiosmosis process. When the thylakoid membrane is present, the second factor is a proton pumping also important. Proton pumping is possible from lumen to stroma. For pumping of protons, the third factor is important. What is it? Proton gradient is important. In the sense, concentration gradient has been created. How does it? These protons concentration have been increased from time to time only by splitting of water. By splitting of water is possible by absorption of water through the root system as well as absorption of light with the help of sunlight and activation of chlorophyll. When these 3 factors are available, splitting of water is possible. When the splitting of water is possible, concentration of protons within the lumen have been increased from time to time. Then proton gradient also created. At last, ATPase enzyme is activation is also important. When the protons are moving, ATPase enzyme is activated. Then at last, ATP formation is possible as well as NADPH2 formation is possible. With the help of these 2 high chemical energy, dark reaction is going on. Without light reaction, there is no dark reaction. Because of light reaction is the first phase of that photosynthesis process for the formation of
chemical energy, which is ATP and NADPH. 2. When the energy is present, only the dark reaction is possible in the presence of carbon dioxide.
From this light reaction as well as chemiosmosis hypothesis, there is a few questions were asked in the previous NEET question paper. First question is, which of the following is not a product of light reaction or photosynthesis? In the photosynthesis, which is not produced during the light reaction. Option A, NADPH. In the sense, nicotine, amide, adenine, dinucleotide phosphate, it is a hydrogen ions added. Second option is NADH, nicotine, amide, adenine, dinucleotide. Third one is ATP, fourth one is oxygen. In the light reaction of photosynthesis, NADPH to formation is A, ATP formation is A, oxygen formation is A. But NADH2 formation is generally takes place in the respiration process, not in the photosynthesis. So option B is the right one. So the second question is, water vapor comes out from the plant's leaf through this stomatal opening. Through this same stomatal opening, carbon dioxide diffuses into the plant during photosynthesis. Reason out means, you know that stomatas are present. In the stomatal opening, carbon dioxide is coming inside, at the same time water is evaporates outside. How? Reason out the above the statement using one of this following option. Above this process happens only during night time. No, photosynthesis takes place during night time as well as daytime, but daytime more, night time is less. Then second option is, one process happens during the day, one is happening in the light time. It is also not correct option. Both the process cannot happen simultaneously. Both the process happen simultaneously because of during the daytime light reaction is possible as well as photosynthesis is possible as well as transpiration also possible. So both processes can happen together because of diffusion coefficient of water as well as carbon dioxide. So option 4 is correct one. Because of carbon dioxide diffuses, at the same time water evaporation also possible. Because of option 4 is the right option. So third question is, in a chloroplast, the highest number of protons are found in. Protons are found in. The question option is, one is given as intermembrane space, stroma, antenna complex. The fourth one is lumen of thylakoid. Always the greater number of protons released from the splitting of water molecule, the greater number of protons are seen in the lumen of thylakoid, in the sense inner space of the thylakoid. Question number 4 is, in the photosynthesis, the light independent reaction takes place. What is meant by light independent? Dark reaction. Light dependent, light reaction. Light independent, dark reaction. So the dark reaction takes place in photosystem 2, stromal matrix, thylakoid lumen, and photosystem 1. Photosystem 1, photosystem 2 comes under light reaction. Then here stromal matrix takes place. In the stromal matrix, light independent reaction takes place because of there is no need of chlorophyll molecule. Stromal matrix is a site for light.